cardiac cell contains bundles of protein strands called myofibrils. These myofibrils are surrounded by sarcoplasmic reticulum, which contains cisternae, dilated terminals. The sarcomeres are the contractile unit of myofibrils, and the T-tubules are continuations of the cell membrane located near the Z lines, which conduct the action potential up to the interior of the cell. The T-tubules connect the sarcolemma to the sarcoplasmic reticulum in the skeletal muscle and the cardiac muscle. Cardiac muscle is an involuntary striated muscle, which is mononucleated and has cross-striations formed by alternate segments of thick and thin protein filaments, which are anchored by segments called Z-lines. Cardiac muscle is relatively shorter than skeletal muscle, and actin and myosin are the primary structural proteins. When the cardiac muscle is observed by a light microscope, the thinner actin filaments appear as lighter bands, while thicker myosin filaments appear as darker bands. The dark bands are actually the region of overlap between the actin and myosin filaments, and the light bands are the region of actin filaments. The thinner actin filaments contain two other proteins called troponin and tropomyosin, which play an important role in contraction. Cardiac muscle also contains dense bands, specialized cell junctions called intercalated discs that separate individual cells from one another at their ends, and these discs consist of a transverse and a lateral portion. The transverse portion of the disc acts as its own of firm adhesion and a route of transmission of contractile force, and the lateral portion of the disc acts as a gap junction across which propagation of electrical impulses between the adjacent cardiac cells occurs. This in effect allows the individual cells of the heart to act as a syncytium. Left ventricle, LV. 1. Made of an inlet portion comprised of mitral valve apparatus, subaortic outflow portion, and a trabeculated apical zone. 2. 3 times thicker than the RV and most muscular. 3. Thickest towards the base and thinnest towards the apex. 4. LV free wall and septal thickness is 3 times the thickness of the RV free wall. 5. Mitral and aortic valve share fibrous continuity. 6. LV apex is relatively less trabeculated than the RV apex. Right ventricle, RV. 1. Comprised of inlet and outflow segments. 2. Inlet extends from tricuspid annulus to the insertions of the papillary muscles. 3. Apical trabecular zone extends inferiorly beyond the papillary muscle attachment toward the ventricular apex and halfway along the anterior wall. 4. Outflow portion, conus, is a muscular subpulmonary channel. 5. Arch-shaped muscular ridge separates the tricuspid and pulmonary valves. Right atrium, bra. 1. Thinnest walls of the 4 chambers. 2. Forms the right border of the heart. 3. Gives off the right auricular appendage. 4. Receives the superior vena cava, inferior vena cava, and coronary sinus. 5. Discharges into right ventricle through the tricuspid valve. Left atrium, LA. 1. Forms base of the heart's posterior surface. 2. Gives off the left auricular appendage. 3. Receives 2 right pulmonary veins, sometimes 3, and 2 left pulmonary veins, sometimes 1. 4. Discharges into the left ventricle through the mitral valve. Ventricular septum. 1. Intracardiac partition having 4 parts: inlet, membranous, trabecular and infundibular. 2. Divided into muscular and membranous septum. 3. Membranous septum lies beneath the right and posterior aortic cusps and contact mitral and tricuspid annuli. Atrial septum. 1. Composed of interatrial and atrioventricular regions when viewed from right. 2. Composed of entirely interatrial regions when viewed from the left. 3. Interatrial region is characterized by fossa ovalis. 4. Atrioventricular portion separates the right atrium from the left ventricle.